take a minute and talk about cell transport. We talked about with photosynthesis and cellular respiration that cells have to take in, well, for cellular respiration, cells have to take in oxygen and they have to take in glucose in order to perform cellular respiration to get energy. Well, you might ask yourself, how does the cell take in the glucose? How does the cell take in the oxygen? Well, this is called cell transport. And we know just by basic cell organelles, the cell membrane plays a pretty important part in the cell. Whatever comes in and out of the cell has to pass through the cell membrane. So let's look at the purpose of the cell membrane. Purpose of the cell membrane is to provide the cell shape, support, and protection. Okay, and we talked about that with plant and animal cells. Okay, whatever the shape the cell membrane is kind of the shape that the cell takes. It also provides that stability, that support, and it also protects the cell. Now, the cell membrane is selectively permeable. Okay, the cell membrane is selectively permeable. And what that means is that the cell has tiny openings, this little balloon here, and I'll just go ahead and close that out. The cell has tiny openings that allow only specific particles to pass through it, okay? If you look at the word selectively, okay, it kind of has the word select in it, okay? That means I'm going to pick and choose a select few things, okay? It doesn't allow everything in there, it allows only certain particles to pass through. So that's what it means to be selectively permeable. Now, particles move through the cell membrane in one of two ways. Okay? One way is called passive transport. Passive transport requires no energy whatsoever. Okay? Items come in, they come out, it doesn't use any of the cell's energy. The other way that particles can move in and out is through active transport. And if you remember the word active, remember you're moving around, you're being active, you're using energy. Same thing with a cell. In active transport, energy is required in order for materials to come in and out of the cell. I want to talk a little bit about the experiment. On day 3, you were taking your egg and soaking it in water. Now, what you should have seen in day 3, before that your egg was soaking in vinegar. So when you took it out, you should have, or I'm sorry, it was soaking in water. You should have seen that your egg swelled up, okay? Now, if your egg cracked or something happened, then you might not get these results. But what happened is when your egg soaked in water, it should have gotten bigger, okay? It should have had a larger circumference than when it was soaking in vinegar. That's what you should have noticed. Now, the next day, well, on day 3, I asked you to predict what would happen if you put it in salt water. Okay. Now, what actually happened, or what should have happened on day 4, when we took the egg out, what you should have noticed is that your circumference, or the actual size of the egg, should have shrunk. Why did this happen? Well, in class, we talked about it's because what happened was inside of the egg, all the water was inside. And that water inside the egg had a very high concentration. Okay, there was a high concentration of water that was inside of that egg. And what happened, and if you think about it, it makes sense. When we eat salty foods, we get thirsty, we get dehydrated. So what the salt water did was the salt caused the egg to dehydrate, and that water was needed, okay, and it exited out of the egg, causing the egg to shrink.